receptors and their types. So the first question is, what is receptor? Receptor is a macromolecule having a specific binding site with functional correlates. These are protein molecules whose function is to recognize and respond to the endogenous chemical signals. Drug bind to the receptor and give pharmacological action. Whenever we talk about drug-receptor interaction, two important properties are there. The first property is affinity, while second property is intrinsic activity. This intrinsic activity is also known as efficacy. When any drug binds to the receptor, these two properties have very importance. So the affinity means tendency of drug to bind with the receptor, while intrinsic activity means ability of drug to generate this pharmacological action. If any drug wants to give his action through the receptor, then he must have the affinity, means he should bind to the receptor. While this intrinsic activity may differ. Some have intrinsic activity, some do not have intrinsic activity, but all the drugs which act through the receptor should have the affinity. And depending upon this affinity and intrinsic activity, there are 4 different types of drugs. The first type is agonist. Agonist means this drug have affinity and maximum intrinsic activity. Partial agonist means it has affinity but its intrinsic activity is at a submaximal level. Inverse agonist means it has affinity and efficacy is opposite to that of agonist. Means, for example, if our drug acts on the beta receptor and these beta receptors are present on the heart, they will increase the heart rate. But its inverse agonist decrease the heart rate. Means you can say it gives action which is opposite to the agonist. Last type is antagonist, which is very important type. Antagonist means these are the drugs which bind to the receptor and they block the receptor. They prevent any binding of agonist, which may be agonist, partial agonist, inverse agonist to the receptor. Ionotropic receptors. Ionotropic receptors are considered as the fastest acting receptor. Events occur in this receptor are drug bind to the receptor, they open the channel and there will be ion movement inside the cell. This ion movement is very rapid, so they are considered as the fastest acting receptors. Examples of such receptors are GABA receptor, NMDA receptor, AMPA receptor, NN and NMDA. Next type is G protein coupling receptor, or also known as a GPCR. There are different names of this receptor like heptathelial receptor, 7 transmembrane receptor, or metabotropic receptor. Events occurs with this receptor are drug bind to the receptor. This receptor then activate the G protein. This G protein then broken down into 2 different parts. First part is active part, while second part is inactive part. Active part act either through increasing cyclic AMP or by calcium channel or through ion channels, and there is an inactive part. G protein means GDP or GTP binding protein. GDP means guanosine diphosphate and GTP means guanosine triphosphate. This G protein, when we talk about its structure, it has 3 different parts: alpha, beta and gamma. Alpha part is considered as the active part and beta and gamma part is considered as the inactive part. GDP is present with the alpha part. So whenever there is a drug binding, it causes phosphorylation, and after phosphorylation there is a conversion of GDP into GTP. So when this GTP is formed, the alpha part gets separated and beta and gamma parts get separated. So this alpha part considered as the active part, which gives its actions by increasing cyclic AMP through calcium or through ion channel. So depending upon it, this alpha part is again divided into 3 different types. The first type is alpha S, which activates the adenylate cyclase and increases the cyclic AMP. Alpha I part, it inhibits the adenylate cyclase and decreases the cyclic AMP, while this alpha Q part broken down PIP2 into IP3 plus DAG and increases the calcium. Means we can say that this G protein coupling receptor have both functions, stimulatory as well as inhibitory functions. Enzymatic receptor. Enzymatic receptor, this receptor has two domains, extracellular domain and intracellular domain. Drug bind to this extracellular domains, and after this attachment or after this binding, it activates the enzyme, and this enzyme is present at the intracellular end. The most common example of this enzyme is tyrosine kinase enzyme. So there are certain receptors which comes under this property, like cytokines, interleukins, prolactin, growth hormone. This type of drugs act through this enzymatic receptor. Intracellular receptor or nuclear subfamily receptor. This is the only type of receptor which is present intracellularly, means inside the cell, while all other remaining type of receptors present on the cell surface. Inside the cell means it is present either in the nucleus or in the cytoplasm. So drugs are also classified based on their targets. For example, vitamin A acts on the receptor which is present in the nucleus, and corticosteroids and vitamin D acts on the receptor which is present in the cytoplasm. This type of receptor activates or work based on the gene transcription or translation process, or it interfere with the protein synthesis process. So we can say that this type of receptor takes longer time to activate or to work as compared with the other receptors.